is that that was my first comment, which is we are recording the meeting. So just so you know, I think I'm legally required to tell you that. We will, if you can put any comments, any questions, you can use the chat box. Please address them to everybody so that everybody can see each other's questions, and then Kate will moderate the Q&A at the end of the talk. As I say, this is our first Women's Brain seminar since the shutdown took effect. So we're really delighted to be back online. We do have another seminar confirmed on Monday, June the 8th, with Roberta Brinton. That will be Monday, June the 8th at 4 p.m. I will send out details of that a little close to the time once we have a talk title and a Zoom link. But with that, thank you very much to Ben, and thank you to Kate, who I will now hand off to introduce Ben. Thanks.
Okay, excellent. Charles, can you hear me okay?
I can hear you.
Fantastic. All right, so thanks to everybody for coming and attending the first of our Zoom version of the Women's Brain Initiative seminars series. It's a real pleasure to introduce Dr. Ben Neal today. He's just across town from us at MGH. Ben is an associate professor at the Analytic and Translational Genetics Unit at Mass General, MGH, where he directs the Genomics of Public Health Initiative. He's also an associate professor in medicine at Harvard Medical School and an institute member at the Broad Institute. Ben's work is generally focused on gaining insights into the genetics of common complex human diseases. And as you'll hear from some of the work he presents to you today, he focuses on a broad range of phenotypes, including disease states, as well as other areas, including sex differences. Ben studied at the University of Chicago and Virginia Commonwealth University, where he earned his bachelor's degree in genetics, and then went on to get his PhD in human genetics from King's College in London. He completed his postdoc fellowship back here at MGH in Mark Daly's laboratory. And he's focused heavily on statistical methodology. He's been an international leader in this area, having published hundreds of highly cited manuscripts in top medical and psychiatric journals. He's analyzed genetic data from large-scale studies across a range of disorders, including, but not limited to ADHD, autism, age-related macular degeneration, type 2 diabetes, and metabolic disorders. He's contributed to the development of popular software tools such as Plink, one of the most frequently used packages in GWAS analyses, and he led the design of the exome chip, a genotyping array that captures rare coding variation in a cost-effective manner. So his work has been broadly impacting the field over many decades now. He'll talk to us today about some of his recent work focusing on similarities and differences in the genetic architecture between males and females. Thanks for joining us today, Ben, and welcome to the virtual version of the Women's Brain Initiative here at Brigham.
Great, thanks, Kate. And it's a pleasure to be here, albeit virtually. So I'll dive right in and try and talk you through some of the different analyses that we're doing with UK Biobank, exploring questions around genetic architecture, both as it pertains to heritability of complex traits more generally, but perhaps more importantly, what the relationship is between the genetic architecture and genetic influences on men versus women, and how that kind of, at least what we're seeing thus far in our investigations. So to kick off and to try and orient you and the kind of talk more generally, and please feel free to chime in with any questions as we go, is what we did with UK Biobank. So, the UK Biobank's a kind of tremendous resource. I'll describe a bit about the phenotype data, a bit about the genotype data. But I think the thing that's really critical is that working with these large-scale data sets, so UK Biobank's a cohort of about 500,000 individuals with a variety of different kinds of phenotypes collected, is really a team effort. There are lots of different moving parts to these kinds of analyses, all the way from how we manage and store and kind of distribute the data, both internally within our group, but also, any results or derivatives with the general world. And so that's, kind of Sam's remit on the download and decryption, and also actually the dissemination of some of the bulk bulk statistics that we've made available.
Can you hear? Oh, shit. Did we lose Ben?
Analysis software platform that really helps us scale up the analytic piece. And I'll talk a little bit about the computational side. But as with, I think, kind of any GWAS or any sort of study of any description, genotype matters, and how we work with the kind of rich phenotypic data in UK Biobank is, I think, just as important as how we work with the genotype data, and actually is probably becoming increasingly important as these kinds of large-scale biobanks become available. And so really how we, everything from normalization to, maybe even factor analysis that Caitlin Carey here has been driving, as additional ways of working with the phenotype data is really critical. And then kind of the last step is just like, kind of creating the association analyses and running those sorts of things. And so, it's not feasible, I think, for any one person to have, a kind of complete toolkit at this point in time for all of the different things that one might do with these different data sets. And so I think that kind of team science and working together is a really important part of these projects. And it's just something that I like to spend a little bit of time on because it really is changing the way we science, if you will. And then up top here is Claire Churchhouse, who is the scientific advisor for the Neale lab, and she helps to kind of coordinate and oversee all of the different moving parts and activities. Okay, so UK Biobank, it's this cohort of about 500,000 individuals. It was genotyped on a biobank array. There's tons of phenotype information collected, and there are a lot of opportunities to ask a lot of different questions and try and answer a lot of different open questions about the nature of, at least as it pertains to my area of focus, what the distribution of genetic effects really looks like and how similar or different that is across different traits, whether there's heritability, how do we think about gene finding when we are really now moving to this extremely rich phenotype data set with lots of genotype information available. So, I myself was not involved in any part of the design or collection or data generation for UK Biobank. It is actually simply a testament to UK Biobank's willingness to share and disseminate data and make it extremely accessible to the kind of broader community that really enables all of the work that I'm going to talk about. There's a data showcase that you can browse some of the different kinds of phenotype information that has been collected, but broadly speaking, it falls into sort of two main buckets. One bucket is a series of questions and kind of survey information that they collect at the different recruitment sites peppered across the UK that asks about, things like what medications are you taking, what is your, what diseases do you have, it'll take your blood pressure, there'll be a kind of additional biometric measures, those kinds of things, as well as like things about your diet and your exercise and sort of more of the lifestyle aspects of UK Biobank. And then the other half is really trying to curate the electronic health record information that's collected in part through health episode statistics, and so those are sort of through hospital recruitment kinds of things. But increasingly, they're moving to integrate also GP notes through the NHS. And having a nationalized healthcare system does not actually mean that they have a unified, nationalized IT infrastructure for all of the NHS. That's not the way that system is built at this point in time. It's kind of organically gotten to where it is. But it does at least provide many common references and frameworks by which to try and start to extract and share that kind of phenotypic information about individual health and disease, really, particularly given the clinical nature of that side. So it's a really fantastic resource. And the cohort is it's got overrepresentation of females compared to males. This is probably part of the healthy volunteer effect and the fact that women tend to volunteer for research studies more than men do in terms of the ascertainment. They tried as much as possible for epidemiological cross-sectional ascertainment, but there is a, bias. The cohort is less diverse. It is healthier. Its 5-year mortality from the point of enrolling in UK Biobank to 5 years later is lower than an age-matched cohort cross-sectionally from census data in the UK. But nevertheless, it's still a very big data set, and that does offer a lot of opportunities, even if it is not perfectly representative of the entire population. And here's the age distribution. And so the sampling frame was really from this kind of age of 40 to 70 was what they were targeting. And the thinking was that this was going to be a longitudinal study where, over, the kind of coming decades, we'd see a lot of incipient disease emerge. And so having biospecimens laid down, kind of complements the overall approach of trying to understand a little bit more about this kind of why do people get sick, particularly amongst those kind of age-related diseases, the ones that cause a lot of morbidity and mortality later on in life, like your diabetes or your cancer and those kinds of risks, and really trying to understand that. You can also see another sort of ascertainment bias if you look at the distribution of age here, because you got the age of retirement, and then right around the age of retirement, there's a spike in participation compared to everyone else. And so the kind of demographic shift around a bit because of who's actually available to participate in the research. But anyway, it is what it is, and it is a great data set. There's a ton of phenotypic information, as I mentioned. A lot of it comes from questionnaires that UK Biobank designed and then kind of inquired with respect to their various cohorts, or with respect to the cohort in the different periods of recruitment. Here's one example, which is time spent using the computer, this kind of data field 1080. And you can see the distribution for the UK Biobank participants. Now, if we were in real meet space, I suppose, rather than virtual space, I'd ask people to raise their hand and see who spends an average of an hour and a half or less on the computer, because that's like half the participants. And every time I've done this in front of a bunch of academics, no one has ever put their hand up for less than an hour and a half. Around about maybe 6 hours is when maybe some people are like, that's my computer time usage on a daily basis. And then there are people that hang out until 8, 10, 12 hours a day. Now, time spent using a computer, it's not the most informative phenotype, but it's also not a useless phenotype. But it too carries with it phenotypic QC problems. And so the point that I want to make here is that on the left-hand side, you see the deciles from the minimum to the maximum value that are reported. And this is, hours a day, and you only get to put in hours a day. And so they coded it as, 0, 1, 2, et cetera, et cetera, et cetera. Allowing up to 24, and some people introduced, 24 in terms of the number of hours that they use a computer on a daily basis. I just say that cleaning your phenotypes is actually just as important and useful as cleaning your genotypes, because I don't think anyone's really legitimately spending 24 hours a day on the computer, but that was sort of what they put in. So anyway, just a note on phenotype collection. So how do we harmonize these phenotypes? And it's a good question about what are the different statistical procedures you should be deploying on the phenotypes that you're analyzing and trying to interpret in the genetic side. We make use of this toolkit called Pheasant that was generated out of the University of Bristol. And basically there's a notion of different kinds of phenotypes, and for the different kinds of phenotypes, they get processed in different ways. So for example, phenotypes that are continuous, but have a pile up of response values that are the same, they get curated and basically turned into either a binary or ordinal variable, because if everyone's reporting the same, it's probably more of an ordinal, countable thing rather than a kind of continuous, really smooth thing. And that's, distinct from, say, integer, where the values are specified as integers coming in and they get processed in a similar way. But the kind of upshot of this is that it gets turned into either continuous variables on the left-hand side or some of the ordered categorical variables on, in the middle, with some amount of recoding that happens along the way. So a good example of somewhere we got tripped up and have moved to correct before is actually the number of cups of tea a day that you report. So the way that that was like coded up front was 0, 1, 2, 3, 4, 5. That's all fine. But the question went up to 6+, and 6+, rather than get coded as 6, got coded as 600. So our distribution turned into 0, 1, 2, 3, 4, 5, and then 600, which is not a great distribution to work with for the phenotype side. So we basically did a de facto GWAS of 600 cups of tea, or basically 6 or more cups of tea versus everyone else, is the way that that would, kind of play out. But then, like we catch that and like update it and those sorts of things. And so I just want to stress that as the number of phenotypes that we're looking at grows, so too do the headaches of all of the ways that the phenotypes can kind of go wrong. All right. So that's the phenotype side. The genotype, it's what I would say is relatively typical of the kind of current modern GWAS array approach of you got a bunch of content for common variation to provide a backbone for imputation. You got a bunch of content focused on rarer coding variation because of their high value and leverage in interpreting what the association actually means. You can even focus a bit on rarer variation that's maybe a little bit more population specific. And then you inevitably have a couple of buckets of variants or markers that are people's like pet causes in the genome. I care about this phenotype. This locus is really important to this phenotype. Can we include these variants? Good examples of that are things, that are, GWAS hits from the previous scans that we put in to try and just make sure that we have good coverage of variants for replication of the data. Or similarly, regions of the genome that are of interest, like ApoE or the HLA or some of the other loci where people really think that there are important associations or biological functions that are already documented, and as a result, having really good information there is really important too. Okay, so that's the array. So we've been working with UK Biobank data now for, I guess, about 2.5 years, almost 3 years. And when it became clear that they were going to release, the big cohort and kind of line it up, it seemed like the right idea at the time to just generate genome-wide association summary statistics systematically across every phenotype that was available in the cohort. And that initial analysis taught us a lot, but probably the thing that it taught us most prominently was that like putting readmes about interpreting your association statistics summary files everywhere on your website, like literally everywhere on your website, is one of the most important things to do up front, because it'll help keep you sane, because that was by far the most commonly asked question. But there were a number of other things that people flagged, like, what covariates do you want to include? And maybe can we do a little bit better in terms of expanding the sample size? And so we took some steps to address that. But I just want to flag that all the way through this project and this series of projects and series of analyses that I'm going to describe, we have, where possible, released summary statistics in real time, pretty much immediately once we were reasonably satisfied that they were of some value more broadly. In a sense, mainly to follow the lead that was already laid down by UK Biobank. So how do we do it? Well, I mentioned HALE a little bit earlier. The main model for how we approach this computation in the analysis, and I will get to some of the things related to sex differences eventually, but just to flag what we're doing on the kind of computation side, we take this big set of GWAS and we actually break it up into as many small GWASs as possible. So we would batch sets of 100 phenotypes at a time. Run one linear regression model on all 100 phenotypes simultaneously based on, basically the like memory constraints of the machines that we were using, and then disseminate it using the cloud and this software architecture platform called HALE. And I think it is likely that these kinds of very rich, large-scale data sets are going to become increasingly common. And at least to my eye, the cloud environment is the only thing that provides us a place where we will be able to actually perform the analyses that we want to perform in a reasonable time. Now this looks great, right? You know, I've just taken the 10 compute hours and broken it up into, you know, 10 one-hour chunks that we've disseminated. The problem is nothing in life comes for free, and what it, you know, the price we pay here, the additional cost, the marginal increase in cost is sort of the overhead of deploying the data and analyzing it jointly across many different places simultaneously. And so it is not quite as, you know, necessarily efficient on the number of computations that you're performing, but I'll take a 10% tax for, you know, order of magnitude reduction in the amount of time it takes to run the analysis. And in point of fact, we can run these kinds of GWAS on, you know, thousands of phenotypes in about a day, which is, you know, I think meaningful because it allows us to do it more than once. Okay, so let's go into some of the things that, you know, we're looking at, some of the ways that we're thinking about what is in there and what's not in there, and then we'll go into a deeper dive about some of the sex differences work. So here's the GWAS of, are you taking cholesterol lowering medication, yes or no? So this is just survey data from UK Biobank to try and give you a flavor of what's going on. And we can contrast that with the GWAS of lipid levels that has been published from the Global Lipids Genomics Consortium. And, you know, we see a lot of loci that overlap, you know, like the clusters over here and some of the clusters over here, like the apo cluster overlaps. And so it's sort of, it's like, what do we learn? What do we don't learn? Well, you know, oops, I don't have the genetic correlation here. The genetic correlation that we estimate between LDL cholesterol from the Global Lipids Genomics Consortium and are you taking cholesterol lowering medication is around 0.5. And a genetic correlation, I'm going to talk a lot about genetic correlation in the rest of the slides. So if you do have questions, please do ask them. But the genetic correlation of 0.5 points to a lot of shared genetic architecture between whether or not you say yes to the question of are you taking cholesterol lowering medication and measured lipid levels in the population, which is good. But there's also a lot of residual variability. And I think that's going to be very much the rule rather than the exception, particularly when we get to large enough sample sizes to start to try and probe what's shared and what's specific. So I think these kinds of simple questions of, you know, what medications are you taking? What can you tell us about your health and well-being, and linking that with genotype has some value, and it definitely bears on this question of the GWAS of lipid levels, but it is not a perfect proxy. And so I think, you know, kind of both kinds of information are the kinds of information we should be trying to synthesize across, because UK Biobank's a very big data set, and this was a very easy question to collect. And so even if it's not a perfect representation of the phenotype that you really are interested in learning about, what you can bootstrap yourself forward with, you know, from these kinds of questions is still potentially valuable. Okay, so why did we do a second round? Well, there were a bunch of SNPs that were imputed in the first go round that were then flagged as not being imputed well, and so we excluded them from round one, but we didn't actually go back and backfill them in round one, so we added them in round two. But we also expanded the list of permissions for even more phenotypes, but perhaps most importantly, we got a lot of feedback on improvements for the GWAS. And there were questions around including age as a covariate. That was the most frequently asked science question, is why didn't you include age as a covariate? And complicated feelings about that, but nevertheless, it was the thing that was requested most. And then also this question about sex, about whether or not we'll be in a position to start to both analyze the cohort separately in males and females, and, you know, interpret what the genetic architecture looks like across males and females. It is worth stating explicitly for the purposes of this talk, when I talk about, you know, men and women and males and females, what I'm talking about is the kind of sex assigned at birth, sometimes referred to as biological sex. This isn't touching on questions of gender identity or those sorts of questions. And, you know, we've really focused on individuals whose self-report sex and biological sex match. And so that's really the limitation and restriction that I would say on that. And so please excuse any kind of loose language around that. That is what I intend to mean as I am describing these things. So in addition to tidying up what phenotypes we could look at and including some additional covariates in the analysis, we also added a bunch of variants, and we tried to capture more individuals. And so, to kind of tackle another complicated set of issues, who do we include in the GWAS when we're kind of creating this, relatively speaking, homogeneous population from a population genetics point of view? So white British is the terminology that's typically, that was used by the UK Biobank. And the way that they asked about this, the way they arrived at this label was they asked what the ethnic group was with these different responses on the left-hand side. And then they asked this ethnic background question of British, Irish, or other white background. It is worth noting that some individuals changed their response to ethnic background between British and Irish across different waves of the phenotype assessment. And some of these other things do move around a little bit as well. Similarly, there are, they took the kind of genetic ancestry piece, put these labels on top of the genetic ancestry piece, and then defined a cluster and excluded outliers across different parts of the PCs. So what we did was, we took a look at that, and here's the principal component analysis. So this is, your typical GWAS common variant PCA to look mainly at ancestry. And this teal color is where in the white British ancestry subset are. And then the green X's are where the self-reported ethnic background of British really is. And you can see that there's a fair amount of, at least ancestral diversity represented in the individuals that report the ethnic background of British. So what we did to try and, I guess, move away from those labels and move from a kind of slightly more genetic ancestry perspective was take the mean and the standard deviation of the top 6 principal components, and then basically drew an ellipse for that entire space. And then we discarded anyone that self-reported as a non-white ethnic background. And that boosted our sample size to about 360,000 individuals versus 337,000 individuals. Now, we focus on this group because there are a lot of different methods that I'm going to talk about that rely on the LD information, particularly the LD score analyses. And so kind of focusing on a reasonably homogeneous group from a genetic ancestry point of view is important. There are other ongoing efforts now to kind of create the multi-ethnic systematic analysis across as many traits as possible, and you should see those released shortly. Okay, so what other covariates did we include? Well, hearing the feedback about age and sex, we included those as well as age squared, and then the interaction terms for all of those as covariates in the analysis, and then we increased the number of principal components. So we're really trying to control for potential confound on ancestry much more aggressively. And we ran it on just the GWAS sample rather than the global sample to really focus in on the cohort that we were looking at. Okay, so all told, that gets us to about 10,000 different GWAS that we've run, because we've got somewhere around 4,000 different traits that are, some of which are sex specific and sex stratified. Actually, this got to an interesting conversation about how we define a sex-specific phenotype. We actually define it in this instance pragmatically based on few, I think something like fewer than maybe 10 or 50 respondents from one of the two sexes as a sex-specific phenotype. It gets a little bit complicated because in the UK, for example, there are part of the phenotypes collected also include occupation. And one of the occupations that was listed is dinner lady. Now, for the Brits in the audience, dinner lady is like a very approachable term. For the Americans, dinner lady is a very confusing term. But basically, dinner lady is like a cafeteria worker in a school. And that's really what you mean by a dinner lady. And all of the dinner lady respondents were women. That was all. Everyone that endorsed dinner lady was a woman. I don't think that means that only women are cafeteria workers in the UK, but it just does illustrate some of the challenges, both in terms of nomenclature, but also in terms of what you consider a specific phenotype as we try and start to think about some of those things. But in any event, that's why there is not 12,000 GWAS, but really 10,000 GWAS, because some of them either just don't make sense in men or women, like ovarian cancer in men is an impossibility, whereas others, there was just no endorsement, even though we didn't necessarily think that it was, prescriptively so. Okay, so let's talk heritability. Well, so, I got this equation here. Let me just spend a minute on the equation. What this equation is an estimator of heritability. So this is the kind of core LD score equation. This L sub j represents the LD score of the variant. So this is the kind of sum of R squareds between the variant that you're looking at and all of the other variants around it, or that is to say how many variants on average that variant tends to correlate with. And then this H squared G is the sort of per SNP heritability. So the H squared G is the whole genome wide heritability, and M is the number of markers. And so you're basically adding a dose of per SNP heritability for every variant that you tag, is the kind of intuition. And then this N is the sample size. So the thing we're modeling here on the left-hand side is the test statistic for the SNP. So, the overall logic is the more variants you correlate with, the higher the probability that you correlate with a variant that is actually influencing the phenotype. And that variant will add, every variant that you correlate with will on average add a, 1 SNP's worth of heritability. Even if we don't necessarily believe that all SNPs are influencing a phenotype, it's pretty robust as long as there's some amount of polygenicity. So, maybe 1 or 5 or 10% of SNPs are tagging a causal variant is probably enough for this machinery to work pretty well. Then we have this NA term here, which is just a sort of intercept, soak up, error, model misspecification, population stratification, other sources of bias that may live in this NA term there. And so this equation now gives us an estimator of heritability, this H squared G, and it's actually pretty trivial to take this from a single univariate case, so one phenotype estimate the heritability, to a pair of phenotypes and not only estimate the heritability for each phenotype, but also estimate the genetic correlation. Okay, so let's talk about heritability, old analysis versus new analysis. So we added more people, but we also added more principal components, and we changed our approach a little bit. What happened? Well, the intercept term, which is really there to soak up the noise or the bias in the distribution of test statistics, that goes, shows less significance with our updated analysis. So we're doing a better job of controlling for confounding or tidying up our GWAS overall. It matches the LD score model a little bit more effectively. So you can see these points below the X equals Y line. So we feel good about that. And then on the right-hand side, it's now the estimate of the strength of evidence of the heritability. So, we add more people, we control our GWAS better, but we also see, on average, a little bit stronger evidence for heritability. And that's consistent, I think, at least in our minds, that our overall analysis is slightly better and slightly cleaner than it was in the version 1. But let's talk about, this was a lot of build up, 20 minutes or so of build up to the thing that I think is most relevant to this audience. And that's about looking into this question of sex-specific effects. Now, we're focused here on running GWAS on males versus females. There are a few natural questions that we can ask in that, frame. The first is, are there clear differences in heritability between men and women for a given phenotype? That could point to a whole bunch of things that we'll talk about. And then similarly, removing the question about heritability, similarity versus dissimilarity, what does the genetic correlation look like across the sexes? So is it the case that the common variant autosomal genetic influences are largely distributed similarly or differently across men and women for many different phenotypes, including things like height and BMI, but also exercise, but also, diseases or lifestyle outcomes, the kind of full array of phenotypic information that's available in UK Biobank. We can ask that question systematically. And that gives us the opportunity also to start to think about probing some SNP level differences. I don't go too deeply into that in this talk, but there is some possibility in kind of exploring that line of questioning. And then, kind of finally, we can take a bit of a deeper dive on anything that is showing any sort of difference. Okay, so first and foremost, what does the distribution of heritability look like overall for all of the phenotypes that we analyzed where we think we're reasonably well powered, at least with a kind of sufficient and effective? And on the left-hand side, you can see the histograms of the point estimates of heritability. And then on the right-hand side, you can see the p-values associated with those estimates of heritability. By and large, the average heritability is around 10%. Now, I don't recommend interpreting heritability estimates super aggressively. I think the important, in terms of the precise value per se, I think the important thing to take away from this is that there is some clear evidence of the contribution of common genetic variation to most of the different phenotypes that we're looking at. And actually, we have a devil of a time really ruling out that there's any heritability whatsoever for any given phenotype. But overall, the distribution looks pretty similar between men and women. And here it is in bivariate form, looking at the point estimate of the heritability in women versus the point estimate of the heritability in men. And what we can see is that there's a pretty high degree of consistency in those point estimates. Raymond's done a great job highlighting the kind of different phenotypes where there may be some evidence of difference, not so many. We're seeing 10% of traits having a nominally different kind of heritability between men and women compared to our expectation of maybe 5% with our p-value threshold, 0.05 for that nominal significance. And then there are a handful of things that surpass our kind of experiment-wise correction, and they're laid out here. So you see 2 blood pressure phenotypes. We have tried to understand what is going on with the heritability and blood pressure. So both systolic and diastolic blood pressure are showing different estimates of heritability in men versus women. Now, I said let's not scrutinize them too much, but this is now the same machine measuring the same phenotype on the cohort, approximately speaking, showing differences in the contribution of common genetic variants. And so this is, one of those instances where I think trying to interpret it does make sense. We see higher heritability in females versus males. And we tried to look at medication effects, so self-report antihypertensives, since, I think men tend to get prescribed antihypertensive medication more than women do, and hypertension is more of a problem amongst men than it is amongst women more generally. That didn't account for any of the sort of material differences if we excluded individuals that were reporting being on the antihypertensive. So we don't actually know what it means. I mean, it might point to there being more measurement error in the male kind of phenotype than there is in the female phenotype. It could also point to different sources of environmental variance. So there may be environmental exposures that are more common in men influencing blood pressure than they are in women, and that those aren't really being heavily genetically mediated. Or it could be that these are 2 complex traits that have different compositions in terms of the relative weight in getting to blood pressure amongst men versus women. And what we're seeing is a pull through in the different heritability of the upstream risk factors that are influencing the downstream phenotype measure that we see here. So for example, drinking behavior may be a bigger slice of the phenotypic variance in, blood pressure than it is in men than it is in women. And if drinking is comparatively less heritable than, say, some other signaling or homeostatic process that is related to maintenance and regulation of blood pressure more generally in humans, that's more prominent in women, that might be a little bit more heritable. Honestly, we don't know. It's just worth flagging. For some of the other phenotypes that we see, the heritability of beer plus cider intake, it shows, higher heritability in men versus women. I think this is heavily contaminated by the fact that, this doesn't look quite as extreme when you look at total alcohol intake. I think it's really the restriction to beer and cider versus, say, all spirits or all form of alcohol, where the heritability does equilibrate a little bit, although it is still admittedly a little bit higher in men than women, even doing that. But I think, you know, there's a pretty significant difference in the rate of beer and cider intake in men versus women, and it's probably a better proxy of overall drinking behavior in men than it is in women. That the women that aren't drinking beer and cider might be drinking wine, but that's not really being captured by that line of questioning. And then we have a few other things that are, you know, I think, you know, whenever I show the number of operations self-reported and the fact that women show higher heritability than men, the natural inclination there, I think, is that it's like measurement error, that men are less reliable and less accurate at reporting the number of operations they've had compared to women. But that's, you know, I guess very speculative, and we don't necessarily know that. And then this duration of vigorous activity, which is one of those exercise questionnaires. So it's a kind of lifestyle-y thing. But I guess, you know, zooming out again, by and large, we're seeing actually a high degree of consistency in the heritability between men and women. And that I think is actually, you know, somewhat meaningful and interesting and important. Michel Navard and Matthias van der Zee, you know, kind of probed this a little bit further by looking at a GWAS of males versus females that Andrea Gana and John Perry and others also picked up and helped out about, and you can read about it on this bioRxiv paper. But you know, fundamentally, if you do your GWAS of are you a man in UK Biobank versus are you a woman in UK Biobank, you run this GWAS, it looks pretty flat, but you do get a heritability. You get a heritability of about 1%. And that's, I think, interesting because these are common genetic variants that are slightly more common in men versus women, or slightly more common in women versus men, that are sort of scattered across the genome with a polygenic architecture, where LD score is, you know, it's not picking up a massive heritability, but the analysis is pretty well powered. And so it kind of poses the question of, well, what's going on here? And the interpretation that I think makes most sense is that what's going on here is actually differential ascertainment bias. So if we imagine a scenario where the individuals that participate in research studies are influenced by heritable traits, or at least partially influenced by heritable traits, those heritable traits will come out as being contributors to the, you know, differences between the groups. So to kind of maybe make that a little bit more concrete, what we're trying to do is estimate the genetic correlation between the heritability of the male versus female GWAS all across the autosomes. So we're looking at every common variant site, all, you know, on chromosomes 1 through 22, so, you know, ignore our sex chromosomes. And we're asking, what are those genetic influences correlating in terms of the kind of heritable component, right? Now, if the reasons for participation were completely random, we would not expect to see heritability in the analysis of men versus women. So even if the overall ascertainment is biased, but it is not biased differentially between men and women, this analysis won't work. So this is actually pointing to differential ascertainment bias, or differential survivorship amongst individuals that would be brought into this study. And what we see here are some phenotypes that are, in a sense, correlating, at least the genetic correlation between ascertainment and what's going on is starting to show up. So here we have a negative genetic correlation. And what that means is that individuals that are male tend to be higher than average, or individuals that are female tend to be slightly lower than average from a genetic landscape point of view on traits like how much time you spend watching the TV or neuroticism. So high scoring females that score high on neuroticism are less likely to participate in UK Biobank than males that score highly on neuroticism is sort of the way to interpret this. It's not a guarantee that there's a causal relationship between the neuroticism and what we're seeing. It's just correlating. So we're just picking up on that correlation, but it's a kind of indication of what might be influencing things over there. So these look more like, you know, potentially, neuroticism things or some BMI or health-related things, where women that have higher BMI tend to be slightly underrepresented in this cohort. It is not necessarily, again, acting through BMI. It is just what we're seeing the correlation with. And then on the right-hand side, we see the absence of high males or low females. And so this is to say, men that are very high on this particular phenotype are less likely to participate than women that are very high on this phenotype. And from a genetic loading point of view, not from a phenotypic point of view. And they include a lot of the things that we might think of as externalizing behaviors, things like risk-taking, cannabis use, alcohol use, et cetera, as well as some relationship with, say, education and IQ as measured in UK Biobank. Okay, so we can do a GWAS of males versus females, or really you can do a GWAS of one group versus another group, and that will give you things that relate to the group membership, but the group membership may be a consequence of ascertainment bias, or maybe a consequence of biology. So all of these things are sort of baked in at some level. It's just males versus females, I think, provides a very convenient way of thinking about what's going on there epidemiologically. But now I'll sort of spend the last, say, 5 or 10 minutes or so talking about the male-female genetic correlation. And so we use the same LD score toolkit to try and get at what is the nature of the relationship between men and women as it pertains to genetic correlation. And here's the overall distribution of female to male genetic correlation across all phenotypes in UK Biobank. Here's the distribution of tests for tests that it's not equal to 0, so pretty strong evidence. There are a handful of points here in the bottom end, the low end, that I'll highlight. So the lowest point estimate of genetic correlation is a phenotype of what is your preferred type of cereal, bran, yes or no? Okay, so it's like bran cereal versus all other kinds of cereals. This, as you might imagine, is not a particularly heritable phenotype. And as a result of being not a particularly heritable phenotype, B is very noisily estimated. So this is not a particularly well estimated kind of genetic correlation that's distinct between men and women, but the data are what they are, and it's good to present them in as unbiased way as possible. Second down on the list is diseases of the urinary system. Okay, so that maybe has a little bit more face validity than choice of cereal, brand, yes or no. I think it's fair to say that, broadly speaking, the different anatomies yield different reasons for diseases of the urinary system. And those anatomical differences are showing up in the contribution of autosomal influences across the genome. And that's very similar to the next lowest one, which is on hernia, which again comes largely from anatomical differences between men and women, and also kind of hernia. So men tend to get inguinal hernia or hernia of the groin, and that is not going to happen so often amongst women. And so again, the phenotype itself has a kind of fundamental different structure. Now, the mean of this distribution's about 0.91, so it's a shade below 1. It's actually, quite clearly below 1, and so we can actually, kind of turn the question not from what has the kind of biggest gap in genetic correlation between men and women to what has the strongest evidence for differences in autosomal contributions to the kind of overall heritability in men versus women. And so now conditioning our null on assuming a genetic correlation of 1, what are the phenotypes that show the kind of strongest drop from a genetic correlation of 1 between men and women? And top of the heap is trunk fat percentage. And actually a whole host of fat percentage phenotypes are collected in UK Biobank, like left arm fat percentage, right arm fat percentage, et cetera, et cetera, et cetera. Those phenotypes really do show clear evidence of dimorphism as it relates to the common variant influences. And indeed, the giant folks, particularly prompted by Cecilia Lindgren and Ruth Lewes, had already showed that waist-hip ratio corrected for BMI, which is thought to at least be some proxy measure of fat distribution, also was showing some clear heterogeneity, not only genome-wide, but actually particularly at the locus level for the influences of common genetic variation on those traits. So we do see some evidences of differences there. So that captures the trunk fat percentage, the kind of hip and waist circumference, and things like weight. There are sort of lifestyle things, or maybe more heavily, another way of describing them might be culturally conditioned things. So things like age of first sexual intercourse or lifetime number of sexual partners, also collected in UK Biobank, a measure of kind of risk, partly related to risk taking, and what we might think of externalizing disorders in the kind of psychiatry universe. Those show some pretty substantial differences between men and women. And I think, to kind of speculate a little bit, I think it is fair to say that those traits that are most heavily culturally conditioned, particularly differently between men and women, are the ones that are kind of popping up in this kind of set of phenotypes. So things like tobacco use, alcohol use, and sexual behavior are showing some differences between men and women. And that's quite in contrast to something like height. So height is really well measured, really easy to capture, and the genetic correlation between men and women is very close to 1. I don't think it's quite 1, but I think it's like 0.98 or something like that. It's extremely high in any event. It doesn't make our significant list, and it's pretty easy to estimate the heritability of height since it performs very well. And it is worth remembering that there is a massive mean difference in height. So I use height as the kind of counter example just because it is not always the case, and we haven't actually documented much of a pattern that is traits that show tremendous mean differences that are necessarily going to have differences in the source of variability in the phenotype in the population. I think it's really more risk factors that are differentially experienced between men and women are going to be part of the story, and sort of like, so that's sort of like what goes in to make your complex trait that seems to be driving a lot more of the differences, rather than necessarily a lot of the innate biological variation that is being played out through the small perturbations that are coming from common genetic influences. So that's some of the lifestyle things. And then the other class of lifestyle thing is this sort of like exercise behavior of, whether you join a gym or some of the readouts that are related to exercise behavior, like your pulse rate, your metabolic rate, hematocrit, and hemoglobin. So those kinds of phenotypes are some of the things that are showing some differences there. But brand serial does make our list, even if it is very unreliable, but you can see how big the confidence interval is there. So it's not something that I think is a super robust finding in the scheme of things. Okay, so that gives you a sort of perspective on genetic correlation across the sexes. So top line result is that basically most common genetic influences are shared across men and women for most complex traits. Those things that show clearest differences are things like fat distribution in one side, and then on the other side, there's the kind of slightly more environmental, cultural, kind of lifestyle kinds of phenotypes that are extremely complex and we don't understand terribly well, but they are what they are. But I also want to show you this UK Biobank genetic correlation browser. You just saw my terrifying email that Duncan Palmer's put together. And so it's actually possible to take any of the phenotypes that we run a GWAS on, and if this were a little bit more responsive, okay, here we go. We can kind of visualize the overall genetic correlation matrices. So this is an overall genetic correlation matrix that exists. And we've also generated, oh, that's now dead. I'll have Duncan fix that link, the risk of doing live demos. But we have the kind of genetic correlation matrix estimated for all of the different phenotypes. And so you can kind of go and take a look at some of the different things that are clustering together. So over here are the kind of coronary artery disease things, the kind of angina pectoris or myocardial infarction, and those kinds of pairs of traits here in this kind of blob of CVD. And that's actually much more broadly splayed out with respect to some of the other things like phenotypes that relate to depression and mood. Oh, I think I got a question. Oh, good. Oh, window is not shared. All right, thank you for letting me know. So you can go to this link and have a look yourself would be my recommendation. Thanks for the heads up. So let's go to the pre-baked tutorial. So the kind of personal favorite for phenotypes to look at in UK Biobank is this phenotype miserableness. Miserableness is defined as, are you ever miserable for no apparent reason, yes or no? It's a kind of very British question, and about 40% of the participants say yes to that question, and a very British answer to a very British question. And that shows, pretty substantial genetic correlations with a variety of other mood-related phenotypes. So things like mood swings, frequency of depressed mood, neuroticism more generally. And again, at this theme of, a bad measure collected on a bunch of individuals can still be potentially illuminating about some part of things that are maybe a little harder to collect, but still worthy of exploration. Okay, so I'll conclude there. So we've run about 10,000 GWAS. You got a sense for, kind of how we did it, what we did, and kind of why we did it. The LD score heritability results show that a huge amount of heritability is observed, like that heritability is observed for a huge number of traits. It's not the only contribution to phenotypic variation. In truth, common genetic variation seems to capture on average around 10% of the phenotypic variance. But, it's not, there are many genetic influences that are influencing a great many different traits out there. And we're only now starting to get a picture of what the kind of distribution of those effects might look like, and only start to get a sense of how they relate to important questions and how they intersect with other important questions, like whether they're shared or specific across men and women. And, for the most part, there's some evidence of sex-specific heritability, but by and large, most common variants are shared across the sexes, with the exception of a handful of traits that I really pulled out and spent a little bit more time going into. And so with that, I'll, I guess, open the floor for questions and comments and see if anyone has anything they'd like to discuss.
Great. Well, thank you so much, Ben. Do we have questions? Kate, do you want to moderate the Q&A? I see there's already one from Shanjan.
Sure, absolutely. So do we just want to have people kind of unmute themselves and ask questions? Or I'm not sure I'm going to be able to see everybody who's...
Yeah, I think it may be hard with, we'd have to scroll down and find
I see.
them.
Got
So
it. I see. Got it. In the...
questions out, that might be the easiest, or Tracy.
Yep, sure. Okay, so it looks like there's a few questions here. How to intuitively understand an inheritability H2 of 10%?
Right. Yeah, I can jump in there. So the first thing and the most important thing to understand and intuit is that this is a property of a population, not of an individual. Okay. So, and the reason that I kind of state that first and foremost is that it is possible to have an estimate of heritability of 10% if some small fraction of your individuals are all completely a consequence of a very strong genetic risk factor, and then everyone else is completely random. That will give you a heritability. It won't likely give you a heritability that looks like the kind of heritability that I'm talking about here, the kind of common variant heritability of many SNPs scattered across the genome that, really hats off to Sean for being one of the big major drivers of that insight at a community level. That kind of heritability, it's more like, some phenotypic variation in the population is correlating with genetic variation. And with LD score regression, it's really being driven by, I think it's fair to say, a kind of polygenic inheritance model rather than some of the other different flavors of heritability that might be out there. In general, it sort of means that, you know, some fraction of the variability in the phenotype that we're observing is a consequence of the genetic variation that is observed in the population. And so it is only appropriate to say that the heritability of 10% that we're estimating here, it really only holds for this study. Actually, it's not a general statement about the heritability of the phenotype everywhere. It is one point estimate of the heritability in the population at this point in time. But it does mean that genetic variation clearly contributes to the phenotypic variation through some mechanism. What that mechanism is, you know, I think the kind of $64 million question that's, you know, confronting the genetics community writ large right now, because we have a really hard time kind of dealing with that. So it just means that there's some biological changes that are happening from your genotype. Those biological changes are turning into some kind of phenotypic consequence, and that phenotypic consequence is making you slightly maybe taller or shorter, or drink slightly more or drink slightly less than you would if you'd had a different set of genotypes.
Thank you. I'm going to take the prerogative and ask another question, just sort of not on this list specifically.
Sure.
In thinking about kind of the data that you presented in GWAS and polygenicity more broadly speaking, I wonder if you could comment on sort of next steps and how we can learn from these kinds of broader analyses about mechanisms. So the recent C4 findings from McCarroll's group. I'm wondering if you could kind of comment on that and just how it fits into this kind of work and sex differences.
Right. I think that's a great set of questions. So so a few answers to that. The first is there are lots of different pieces to the sex differences in terms of the kind of overall scientific pipeline, if you will, like the overall scientific landscape. And like in terms of trying to, you know, disentangle certain mechanism things, if we had extremely well-powered GWAS in men and women, then the loci that were showing substantial differences in effect size between men and women might point to slightly more coherent biological entities. So it stands to reason if there's a genetic influence that is specific to men or women, then it's probably related to something that is the source of the differences between men and women. Now, whether that source of difference is like hormone levels or cultural expectations, you know, or anything in between, right? Because that's like the full spectrum of possible sources of difference and variation in the population there, just to kind of frame it. It nevertheless would help to try and, I think, narrow down and aid in the interpretation there. I also think that, you know, it has implications for if we do get good at interpreting the consequences of genetic variance for what they are doing biologically, and can start to turn that into mechanistic insights about specific disease, like you alluded to with Steve's work on C4, then being able to get mechanistic insight into the things that are shared between men and women and the things that are specific to men and specific to women, I think is also super important. But I think we really need to, you know, unblock the interpretation of genetic association identification to be able to kind of coherently do that. C4 had a lot of nice things going for it in that, you know, it's copy number of a gene, and so it took the what gene is it problem off the table and just put the problem back on the table of what does the gene do. And while there's evidence in support of synaptic pruning as the mechanism for C4, I would certainly say, and I'm reasonably certain that Steve agrees, as does Beth and many others, that this is the best working hypothesis, but we are far from definitive proof that excess synaptic pruning is exactly the mechanistic, you know, deficit that's driving the increased risk in schizophrenia in the population. So I guess it kind of depends on where you want to do the work. I mean, in the mouse world, for example, a lot of the work gets done only on men because it's easier and cheaper. and I guess what we're trying to do with the genetics work is at least allow us the possibility of exploring what some of these differences might be and might mean. And I guess that's the direction that I would try and take it in. And then I guess another comment to make is, I believe that what other phenotypes a given SNP is associated to is an annotation of great importance for that SNP. And so having these large-scale systematic evaluations are really, really important because, if I'm focused on one given disease area, I don't always know whatever else this gene might be doing in the biological system. And that has implications for side effects and has implications for, how we think about interpreting those association findings. Okay, so there's a question about sex chromosome that's been asked twice. So we don't have LD score genetic correlation machinery off the ground for the sex chromosome. But we did ask a slightly different question, which is trying to look at what the right estimate is for beta. So for doing the GWAS. So when you do your GWAS of X chromosome, you kind of have two options for how you code it. You can either code, so if we code women as 0, 1, 2, we can either code men as 0, 1 or 0, 2. And there's actually arguments to do both, curiously enough. So that is to say, if X inactivation works really, really well for all the X chromosomes, and there really is no expression, then 0, 1, 2, and 0, 2 might make sense in terms of where we would expect the biological consequence. Empirically, when we look at that, we get a mess, to be completely honest. And actually, it's not clear that 0, 2 or 0, 1 is right uniformly. I actually would say it's pretty clear that it's not right uniformly, and that there are some complexities about what the influence of a given genetic variant is on the X chromosome in men versus women. I would also point you to some work that Barbara Stranger is doing, looking at GTEx, which also explores that set of questions very, very deeply, and I think is also really informative. But I would say where we are right now on the X chromosome, things are complicated. Estimating the heritability contribution is challenging. We're working on, we'll get there. It's just, it requires a little extra nuance. But the SNP association results themselves are actually also quite interesting in that they do point to this kind of different dosage effect in the beta.
Can I ask a question? This is Charles. Ben, I think you just said that, so you're looking only at common SNPs in this population.
Yep.
How much of the genetic variation is going to be missed as a result of not looking at either de novo mutations or copy number variants or whatever else it is that you might expect to see? And what are the prospects for studying those kinds of variation in the future?
So I'll do them in reverse order. The prospects are quite good because UK Biobank is going to get whole genome sequenced. It's already being exome sequenced, but the genome sequencing's planned, and that is going to happen and will be released to the community and the population more generally. Yeah. So that will allow a little bit more of an extensive exploration there. In terms of the other kinds of variation, there are sort of two answers to that question. The first is, if it's a common variant, it will be tagged anyway, even if it's not a SNP, because it's a common variant and it has to obey all of the same population genetic rules that SNP variation does. It may not obey them exactly correctly, but tagging will capture a lot of that variability in general, with some caveats about mutation rate and the breakdown of LB, but by and large, that's the expectation on common variation. And the capture of common variation should be pretty good. The second comment is, the second comment that I would make is that in terms of number of genetic variants, you're right. This is a tiny fraction of all of the genetic variants that are present in the data set. The amount of your genetic variation, given that you are in the data set, however, is the overwhelming majority. Almost all of your differences between everyone else are down to common variants. That rare variants are actually a very, very small contribution to the differences between you and another random individual in the population. It's actually a tiny, tiny contribution in that sense. Now, it can be a little bit enriched for function and for very, very severe phenotypes and Mendelian-like things, then yeah, that's completely off the table. But for complex traits where selection's having a hard time operating on the genetic variation because the genetic variation has a very, very small effect, and so most of the things look like drift, which is more or less where we are right now in terms of how we think about the common variant contribution to genetic architecture. This will do a pretty good job.
Great, thank you. And I have to ask one more question as a British person. Who picked the list of phenotypes? I mean, whose idea was it to ask about preference for brand cereal or beer and cider or miserable?
It's UK. So the UK Biobank has a kind of study team, and it's very heavily epidemiologically mediated and driven. So like public health and epidemiology are the major drivers of the cohort collection. The genetics got bolted on after the fact. It was more, like, hey, we have this big data set. It would be good to kind of genotype it. And so it's, I guess, probably more of a reflection of what the epidemiologists really care about. And nutrition is an important aspect of epidemiology, sort of whether we like it or not.
Great, thank you. So I have another question before I see if there are other people. Oh, I see there are a couple more questions, but I'm wondering if about how you address gender versus sex and sort of the concordance or discordance of biological sex and gender, and if that's in the UK Biobank, can it be looked at, and so on?
So there is a small sample of individuals that are discrepant between their self-reported sex and their, kind of genetic sex, if you will, as well as a handful of sex aneuploidies and other things like that in the cohort. I mean, it's a really big cohort. And there's also mosaic loss of Y to varying degrees that is being estimated, I think, from some of the earlier sequencing data. It's not great because, it's not, this is not asking the questions around sort of gender identity and some of the, what I would describe as more, nuanced approach than just simply focusing on the kind of binary world. I think, I think it's probably not the, it's not collected in a way that I think reflects the ways that the kind of non-binary community and other parts of the kind of LGBTQ plus community might design a questionnaire around gender identity, I would say. And so I think, this is, probably not the right place for that. And I think it is, very much a limitation in terms of what is available. And it's a hard thing because, there are also other reasons why they may be mismatched, like they may be mismatched because there's a sample swap. And, that's kind of a common reason that we might have that kind of problem. And indeed, with some of the earlier discrepancies that were identified, they went and recollected and did discover that it was a sample swap, I believe. So I think there is some precedent there for that. And you can kind of go digging into some of the UK Biobank, like sample QC things in the Bycroft paper that I think talks about some of those issues. And so that's the kind of concern that I would raise about trying to analyze and interpret that side of things.
So there's a question about whether there's another data set you wish you had to better understand sex differences. I will point out before Ben answers this that he has access to a lot of different data sets. but go ahead and answer that, Ben.
I mean, I think, you know, I guess, you know, the biggest comment that I would make is that there are a lot of interesting questions about, I mean, I think the easiest answer to that question, frankly, is if I had access to the individual level data for 23andMe. That would probably make me happiest, because it would allow me to do a hell of a lot more than I can do right now in terms of the way that they've structured it. But I think I use that as an example because I think it is the size of the UK Biobank data set that really enables the most thorough investigation here. And even though we have the most thorough investigation of this question, we're still very noisy. So there's a question a little bit above about how we get estimates of genetic correlation that are greater than 1. Well, the estimator that we use is an unbiased estimator of genetic correlation, which means we can statistically allow the estimate to go above 1, even if we don't believe that it is 1, because the way that we're running the kind of regression and normalizing it against the other 2 regressions. And so if you want an unbiased estimator, you have to tolerate point estimates that are out of bounds of what we would allow, because of the way that it's working statistically. And the fact that, you know, you were seeing in that slide genetic correlation estimates of, you know, 1.4 just underscores how noisy those point estimates really can be, if that makes sense. So the other data, you know, the data set that I wish I had access to, I guess I wish that everyone that did disease GWAS would release summary statistics for males and females separately. I think that would allow for a much more systematic investigation into these kinds of questions. And, you know, this is something that's standard in the animal literature and kind of magically not in the human space. And it is a really big piece. Okay.
Great. Well, thank you. Kate, do you want to have the last word here?
I'll just thank Ben for an interesting presentation and appreciate your flexibility in getting this kind of set up in a virtual manner. Hopefully in the future we can have an in-person visit from you as we had planned originally. If there aren't any additional questions, I'll give a couple seconds. Anybody have any pressing questions? Excellent. So thanks very much, Ben. It was a pleasure hearing the talk. Charles, do you have anything else you wanted to add?
No, just add our thanks. Thank you so much, Ben. Thanks, Kate, for moderating. Thanks to everybody for joining us. And we'll hope to see many of you at Roberta Brinton's talk, June 8th. So stay well, stay safe until then. In fact,
Cheers,
stay
Charles.
beyond then. Thank you.
Take care. Thanks, Ben.
Bye, everybody.